Welcome to the Deep Dive. Today we're doing something really foundational. We're going to dive into the four great classes of biological molecules.
And these aren't just, random ingredients. These are the pillars. They are the essential structural and functional components of every living cell.
Exactly. We're looking at the architecture of life itself. And what's amazing, really, is how all this incredible complexity is built from a surprisingly, a small set of building blocks.
That is the core idea. I mean, if you boil it all down, life has this amazing economy of design. You have these massive structures, these macromolecules, but they're all built using simple, repeatable rules.
Okay, so three of the four classes, carbohydrates, proteins, and nucleic acids, are what we call polymers.
Right, which just means they're long chains made up of smaller, repeating units, the monomers.
I would think of it like a train. The polymer's the whole train, and the monomers are the individual boxcars, just linked together one after another.
That's a perfect analogy, but we have to flag an exception right away.
The fourth class, right.
Yeah, the lipids. They're a huge, incredibly important group of molecules, but they aren't true polymers. They don't form those neat repeating chains. They're sort of the odd one out.
So let's talk about the assembly line. How do cells actually build and break down these massive chains? Because there's beautiful efficiency to it.
There is. It all comes down to two simple opposite reactions, both helped along by enzymes. To build a chain, to link two monomers together, the cell uses a dehydration reaction.
And the name says it all. You're removing water.
Exactly. One monomer provides a hydroxyl group, the OH, and the other provides a hydrogen, the H. They combine, leave as a water molecule, and a strong covalent bond forms between the monomers.
So remove water to build the chain, which means to break the chain, you just
add the water back in. It's called hydrolysis, literally water breaking. The water molecule splits and breaks the bond, separating the monomers.
And that simple pair of reactions, dehydration and hydrolysis, is behind almost all the building and recycling going on in your body right now.
It's the fundamental engine.
All right, let's jump into that first class. Carbohydrates. Most of us just think energy or sugar.
And that's a big part of it, but they're also for structure. The monomers here are the monosaccharides, or simple sugars, and the king of those is glucose.
Glucose is the cell's primary fuel. But I think it's easy to forget, we always see it drawn as this straight line in textbooks.
Right, but in water, in the cell, it immediately snaps into a ring.
A more stable ring structure.
Yep.
And that's its functional form.
Absolutely. And that ring is what cells bore down through cellular respiration to get energy, but its carbon skeleton is also used as raw material to build other things, like amino acids.
Okay, so if we link two of those rings together, we get a disaccharide.
Right, using a dehydration reaction. That specific bond is called a glycosidic linkage.
And a classic example is sucrose table sugar, which is just a glucose linked to a fructose.
Or lactose, which is milk sugar. And this is actually a great example of hydrolysis. If you're lactose intolerant, it just means your body doesn't produce the enzyme, lactase, that's needed to add water and break that specific glycosidic bond.
Let's scale up to the big chains, the polysaccharides. These are for storage and structure.
For storage, plants use starch. It's just a long polymer of glucose. Animals, on the other hand, use glycogen.
Now, glycogen is also a polymer of glucose, but it's much more branched than starch. Why the difference?
The branching is key for function. All those branches give you more free ends on the molecule, so when your body needs a quick burst of energy, enzymes can attack all those ends at once, releasing glucose much, much faster.
So it's about rapid access.
Precisely. Now, for the structural side, we have to talk about cellulose.
The single most abundant organic compound on the planet. It makes up plant cell walls. But here's the crazy part. It's also a polymer of glucose, just like starch.
Yeah, and this is where a tiny, tiny change in structure has massive consequences.
So in starch, the glucose monomers are all oriented the same way, linked by what's called an alpha linkage.
Correct. But in cellulose, they're joined by a beta linkage. And that seemingly tiny difference forces every other glucose monomer to be flipped upside down relative to its neighbor.
And that flip prevents the chain from coiling up like starch does. Instead, it stays perfectly straight.
Perfectly straight. And those straight chains can line up next to each other, forming hydrogen bonds between them, weaving together into these incredibly strong cable-like microfibrils. That's what gives wood its strength.
So here's the punchline for us. Our digestive enzymes can break the alpha linkages in starch.
But they are completely powerless against the beta linkages in cellulose. They just don't fit.
Which is why we can't digest wood or grass, and why cellulose is what we call insoluble fiber.
Exactly. Its function in our diet comes from the very fact that we can't digest it. It helps keep things moving through the digestive tract. It's a structural molecule through and through.
Okay, that brings us to our second class, the rule breakers. Lipids. No polymers here. So what is their one single defining trait?
They are all hydrophobic. They just do not mix with water.
Oil and vinegar.
The classic example. And it's because they're made mostly of long hydrocarbon regions, just chains of carbon and hydrogen, which are very nonpolar.
So let's start with the ones we know best. Fats. Or, more technically, triacylglycerols.
Right. They're built from a glycerol molecule attached to 3 long fatty acid chains, and their main job is just incredible, dense energy storage.
More than twice the energy of a carbohydrate, gram for gram.
Yes. And the difference between a solid fat, like butter, and a liquid oil comes down to the shape of those fatty acid chains.
We're talking saturated versus unsaturated fat.
Exactly. A saturated fat has no double bonds in its chains. They're saturated with hydrogen.
Well,
This makes the chains very straight and flexible so they can pack together tightly.
which is why they're solid at room temperature.
And an unsaturated fat has at least one double bond. Usually it's a cis double bond, which creates a permanent kink in the chain.
Ah, a kink. So they can't pack together as tightly.
They can't, they're pushed apart, and that keeps them liquid at room temperature. Those are your oils.
And this is where health comes in. We hear about trans fats. Those are made when liquid oils are hydrogenated, and some unnatural trans double bonds are created.
And those, along with high intake of saturated fats, have been linked to things like atherosclerosis, where plaque builds up in your arteries. The molecular shape really, really matters.
Okay, next, lipid. And this one is, absolutely essential, phospholipids.
Phospholipids are the architects of the cell. They are similar to a fat, but they only have 2 fatty acid tails.
And the third spot on the glycerol is taken by a phosphate group.
Right, a negatively charged phosphate group. And this creates a molecule with a split personality. You get a polar hydrophilic head that loves water.
And 2 nonpolar hydrophobic tails that hate it.
And when you throw these molecules into water, they do something amazing. They spontaneously self-assemble into a bilayer.
With the heads facing the water on both sides, and the tails all hiding together in the middle, away from the water.
And that spontaneous barrier, that's the cell membrane. It's the foundation of every cell.
The last lipid. Steroids. They're easy to spot by their 4 fused carbon rings.
And the most famous one is cholesterol. It gets a bad rap, but it's absolutely vital. It's woven into your cell membranes to maintain fluidity, and it's the starting molecule your body uses to make all the steroid hormones, like testosterone and estrogen.
Let's move to our third class, the true workhorses of the cell. Proteins. They do almost everything.
They really do. They account for over half the dry mass of a cell, but their most important job is acting as enzymes.
The catalysts. They speed up all the chemical reactions in the cell without being used up themselves.
And to achieve this incredible functional diversity, they use a set of just 20 monomers, the amino acids.
Every amino acid has the same basic structure, right? A central carbon, an amino group, a carboxyl group.
And then the one part that's different, the R group, or side chain. And that R group determines everything, whether the amino acid is hydrophobic, hydrophilic, or electrically charged.
These amino acids link up via peptide bonds to form a polypeptide chain. But the chain itself isn't the final product. It's all about the final folded 3-dimensional shape.
The shape is the function. A protein is useless until it's folded correctly, and that folding happens in 4 distinct levels.
Okay, level 1, primary structure.
Primary structure is just the sequence, the unique linear order of the amino acids in the chain. It's the blueprint dictated by your DNA.
Then we have secondary structure. This isn't the whole shape yet, just local fold.
Exactly. Local coils and folds. You get the famous alpha helix and the beta-pleated sheet. And these are stabilized by hydrogen bonds forming along the repeating backbone of the polypeptide, not the R groups.
Okay, so the backbone creates some local structure, and then the R groups get involved for the tertiary structure.
Now you're at the overall 3D shape of that one polypeptide chain. And this is driven almost entirely by interactions between the different R groups.
So the hydrophobic R groups all huddle together in the core, away from water.
Yeah, that's a major driving force. You also get hydrogen bonds, ionic bonds, and even strong covalent bonds called disulfide bridges that act like staples, locking the shape in place.
And then finally, for some proteins, there's quaternary structure.
This is only for proteins that are made of more than one polypeptide subunit. Think of hemoglobin, the protein that carries oxygen in your blood. It's actually made of 4 separate polypeptide chains, 2 alpha and 2 beta, all fitting together to make one functional unit.
The power of that primary structure, the sequence, is just staggering. A single mistake can be catastrophic.
Sickle cell disease is the textbook example. It's caused by substituting just one single amino acid for another in one of the hemoglobin chains.
One mistake in a chain of hundreds.
And that one tiny change causes the protein to misfold and clump together when oxygen is low. This deforms the entire red blood cell into that sickle shape, causing all the devastating symptoms of the disease.
And since that shape is so delicate, it can be disrupted by the environment, right?
Absolutely. That's called denaturation. If you apply too much heat or change the pH, you disrupt all those weak bonds holding the tertiary structure together. The protein unravels and becomes inactive.
Like cooking an egg, you can't uncook it.
It's permanent denaturation. The protein's shape is lost, and so is its function.
Okay, our final class, the information keepers. Nucleic acids. This is where we get the instructions for building all those proteins.
Right, the central dogma. Information flows from DNA to RNA to protein.
Nucleic acids are polymers called polynucleotides, and their monomers are nucleotides.
And each nucleotide has 3 parts: a nitrogenous base, a 5-carbon sugar, and a phosphate group.
The bases are A, T, C, and G in DNA. And the sugar is a bit different between DNA and RNA, isn't it?
It is. DNA uses deoxyribose, which is missing an oxygen atom. This makes it more stable, which is great for long-term information storage. RNA uses ribose.
These nucleotides link up to form a sugar phosphate backbone. And of course, DNA is famous for its structure.
The double helix. It's 2 polynucleotide strands running in opposite directions. We call that anti-parallel.
And they're held together in the middle by hydrogen bonds between the bases.
And this is the secret to everything. Complementary base pairing. Adenine A always pairs with the thymine T, and cytosine C always pairs with guanine G.
A with T, C with G, always.
This strict rule means that if you have one strand, you automatically know the sequence of the other. It's the key to accurately copying DNA every time a cell divides.
And RNA is typically single-stranded.
Usually, yes. But it can fold back on itself and base pair in certain regions to create complex 3D shapes. That's critical for RNAs that have jobs to do in the cell, like transfer RNA, which helps build proteins.
So tying this all together, how has modern technology just blown this field wide open?
Well, our ability to sequence entire genomes, the complete set of DNA for an organism, has just changed everything. It created new fields like bioinformatics to handle all this data.
And genomics, the study of whole genomes, and proteomics, the study of all the proteins.
Exactly. And together, they've given us a molecular tape measure for evolution.
You mean we are comparing the DNA or protein sequences between 2 species, we can see how closely related they are.
That's it. The sequence of the beta-globin protein in a human and a gorilla differs by just 1 amino acid. Between a human and a frog, it's 67.
The chemistry confirms the evolutionary history.
It does. It can resolve relationships we were never sure about before, like confirming that hippos are more closely related to whales than to other land animals. The molecules tell the story.
So that's our deep dive, the 4 pillars. Carbohydrates for fuel and structure. Lipids, the hydrophobic group for membranes and energy. Proteins, the versatile workhorses whose function is all about their shape. And nucleic acids, the information archive.
It really is a beautiful system. And I think the final thought to leave you with is just how simple the underlying chemistry is to build or break down almost all of this incredible diversity. It really just comes down to adding or removing a single molecule of water.
The entire complexity of life emerges from that elegant, simple rule.
It's an economy of design scaled up to a planetary level.
Thank you for joining us for this deep dive into your sources. We'll see you next time.